Next, I'd like to introduce Jack Lindsey from Columbia University. His field is computational neuroscience. His advisor is Asuk Litwin Kumar, and his practicum was at Sandia National Labs in New Mexico.
Hello, I'm Jack. I'm excited to be talking to you all. This is some work on, I work in computational neuroscience. I think it's a bit kind of, maybe one standard deviation away from what most people here work on, so hopefully I can convey something to you about this field. This is work with Elias Issa, Columbia. Neuroscientists are interested in explaining what neurons in the brain are doing, and in visual neuroscience, that usually boils down to how do we predict how a neuron is going to respond to an image or a video that's presented to a human or animal. And in the past 10 years or so, it's become increasingly popular to do this using deep neural network models. It's kind of the only game in town for explaining neural responses in kind of higher level visual areas. And the strategy taken is to take a big neural network that you've trained on some other task, like often image classification, present it with some stimuli, present a human or animal subject with the same stimuli, and then see to what extent you can kind of linearly map the responses of the artificial neurons to those of the biological neurons, or maybe it's fMRI voxels instead of neurons. And this works pretty well, or at least better than other methods. And not only does it work pretty well, there was an interesting phenomenon observed even 10 years ago that basically the extent to which a given neural network model is capable of predicting, being used to predict neural responses in the brain correlates fairly strongly with its performance on computer vision tasks. So basically, like ImageNet classification performance is kind of predictive of how well a neural network model is going to be able to account for neural responses in the brain. Or at least it seemed like this as of 10 years ago. But in the past 10 years, computer vision has gotten better and better. People are able to classify images and do other amazing segmentation tasks better and better. But this hasn't yielded a corresponding improvement in ability to predict neural data. So after a certain point, kind of pushing task performance on these computer vision benchmarks further and further is not giving you computational models that better account for what's going on in visual regions of the brain. And this is the case across kind of a whole wide variety of different approaches to training computer vision models, whether it's training them on classification or on unsupervised objectives. It's the case kind of regardless of what kind of neural data you're looking at, whether it's from humans, whether it's from monkeys, whether it's electrophysiology or fMRI. So there's this kind of robust saturating effect, and after a certain point, better classification performance is not really telling you more about what the brain is doing in visual areas. And so that raises this kind of obvious question, okay, so if classification performance isn't all we need, what's the next sort of thing? What objective functions can we use to push models to be more like the brain and give us a better understanding of what the visual cortex is trying to do? And so obviously, the brain has to support human or animal behavior. There's lots of visually guided behaviors, and they don't all boil down to object classification. You have to move around in the world and navigate, and you need to know where objects are relative to each other and to you. You need to know their pose and their movement and all sorts of things. And so, kind of intuitively, we'd imagine an important property for the visual system to have is to encode lots of different kinds of visual information in some sort of organized fashion that enables lots of different behaviors. Okay, so that leads me to the sort of quantity that we've introduced that we propose is a useful kind of principle to think about in addition to categorization as a sort of normative objective of the visual stream. So I'll try to walk through these cartoons because it's not intuitive or it shouldn't be clear yet what they mean. So these are kind of cartoon illustrations. The three axes in these, just look at the top row for now. The three axes in these plots are meant to indicate the responses of three neurons to a set of images. And the different colors are meant to indicate different categories. So say like the blue cloud is like the set of all responses of these 3 neurons to a bunch of images of dogs. And then maybe red is cats and green is humans. And so the top left kind of illustration is meant to indicate what we'd call a representation that's invariant to non-class information. So all the responses to dogs are the same for these 3 neurons. Whereas over on the right, there's the responses do carry information about the class. The different color clouds are distinct distributions, but they're kind of overlapping it. So they're also carrying information about other aspects of whatever these fake images are. And the middle row is, column is supposed to convey what we mean by a factorized representation. So here, the responses of these neurons carry information about the category in the kind of XY plane, and they also carry information that distinguishes images of the same category, but in kind of an orthogonal subspace. Here it's the z-axis. And obviously things can get more complicated with more neurons, which corresponds to higher dimensional spaces. So that's what we mean by factorization, the extent to which different kinds of visual information are driving variance in different non-interfering subspaces in neural activity space. And so you can kind of operationalize these ideas mathematically. So if you have kind of a set of images that in which you're only varying, say, one parameter p. So let's say I have a bunch of images, and I'm only varying the position of the foreground object in the scene, and we'll call that parameter the position parameter p. You'd say the representation is invariant to that parameter if basically the variance driven by changing it is small relative to the variance driven by changing other parameters. And similarly, if you have 2 parameters of interest, p and q, you might say that the representations factorizes those 2 parameters if you identify a linear subspace that captures most of the variance driven by varying q by itself, and then look at to what extent the variance driven by p lives in that subspace, and if very little of the variance driven by p lives in the subspace that captures most of the variance driven by q, that's when you'd say these 2 parameters are being encoded in a kind of a factorized fashion. So these are kind of things that you can measure in neural data by looking at a corpus of images that's been presented to an animal while its population activity in different visual regions is being recorded. And so V4 is a lower level visual area, and IT is a higher level, kind of closer to cognition area. And what you find if this was a data set of images that includes images of different types of objects and in different positions, and what you find is that the kind of representations as you move along the visual hierarchy discard information about kind of everything that's not identity of the object and its position. So they're becoming more invariant to these other factors. But also to the extent that they're not discarding it, it's being factorized into these sort of orthogonal subspaces. So that's kind of a proof of concept that this is a useful idea. I'll skip that. Unfortunately, there isn't a lot of neural data with these kinds of the kinds of images that you need to compute these sort of metrics where you're varying lots of qualitatively different scene parameters independently of one another, and then presenting that kind of carefully curated data set to an animal while recording neural activity at scale. People just haven't really done that. So the approach we've taken is to do that, but with neural network models that we then compare to the neural data that does exist. So kind of a slightly roundabout way of asking whether these properties are present in neural data. Okay, so what did we do? We created this synthetically generated data set of images that consists of foreground objects superimposed on a natural background. And then we can independently vary a variety of different qualitatively different scene parameters, like the identity of the foreground image, the identity of the background scene, the position and orientation of the foreground object, the lighting conditions, the viewpoint from which the scene is being looked at. and vary all of these independently. And that allows you to compute those kind of equations I flashed up earlier of factorization and invariance to these different parameters. So that's kind of step one of the workflow, is computing these properties of a bunch of, we have a big bank of like 100 or so deep neural network models, and we can compute these properties with them. And then step two is, for each of those models, we also want to have some measure of how well it fits brain data. And so that's where we go to this approach that I alluded to earlier of taking images that have been presented to humans and animals in neuroscience experiments, presenting those same images to these models, and then seeing how well their responses linearly map to the biological data. And then kind of cross-checking, which models are like more factorized and which models are more brain-like, and so on. We can get a sense of whether these properties are important for achieving sort of better brain predictive models. Okay, so you kind of go through this exercise and you get a bunch of scatter plots that look like this, which are like, okay, here's a bunch of neural network models. Here's like an example of a property we might compute, like how will they factorize the variance driven by moving the camera around from the variance driven by changing the actual scene. And that's like what's plotted on the x-axis, and then y-axis is goodness of fit to these different neural data sets. And this is just an example, but we kind of do this at a really large scale with a couple hundred different models, 10 or so different neural data sets and some behavioral data sets, working across, you have to kind of optimize over the layer that's being used, which layer of the neural network that you're using for the comparison, and then repeat for all the different metrics we're interested in. And I'll kind of skip to the punchline, which is, so what we find is that this kind of factorization quantity is pretty consistently associated with better fits, better prediction of these different neural and behavioral data sets. Whereas interestingly, invariance to these like different scene properties is kind of inconsistently associated with it. So it seems that, so background and, so models that are kind of filter out information about background and lighting conditions tends to be more predictive of neural data, whereas models that filter out information about viewpoint and object pose tend to be not more and sometimes even less predictive of neural data, suggesting that those kind of quantities are things that in these visual regions are information about them is not being filtered out, but is being sort of pushed into it's being encoded in a way that doesn't interfere, is orthogonal to the encoding of other information about the scene that you might be encoding, like object identity. We also kind of repeated this analysis with variance driven by natural movies instead of these synthetic image data sets. So using kind of different categories of movies, either where the movement is camera driven or object driven. And we get, it's kind of the same result, but it's cross-checking on for this different data source. I think I'm running out of time, so I'll just skip to the kind of future-looking section. So an obvious question that this whole kind of analysis raises is, okay, we have this sort of correlational analysis suggesting that factorizing different information about visual scenes is indicative of brain-like models. Can we actually use that to get better models? So can we incorporate that as can we augment our objective functions for training of computer vision models with these kinds of ideas to push them to match brain data better or to perform better at naturalistic tasks? And there's some really cool work going on by another group that we're working with this year that is sort of has positive results in that direction, incorporating factorization like objectives and model training. And there's a bunch of other kind of questions about how all this stuff is implemented biologically that we're also interested in. But I'll stop there. Thanks for listening, and happy to take questions.